In this video, we will discuss about whether this EMT and apoptosis that researchers have observed in the TGF-β treated compliant matrices are dependent on each other or independent to each other. As the heading suggests, they are independent to each other. So this is what they finally conclude of the results that are presented in this figure. So what they did is they first prevent this apoptosis in this TGF-β treated compliant matrices. So how they did it? They did it by two different strategies. First one, they overexpress the survival factors, that is Bcl-xl, or they use the ZVAD, which is a pan-caspase inhibitor, that is they suppress the apoptotic pathways, we can say that. So first they check in the immunofluorescence, and one more thing before we go into the results, that EMT, that the results changes that they usually see after 48 hours, whereas apoptosis signals they usually see within a few hours. So here they are checking the EMT changes. So when they check the EMT changes compared to on the compliant matrices that has the stiffness around 0.4 kilopascal, they are able to see that there is no delocalization of the end cadherins compared to the control, and also there is no overexpressions of this E-cadherin and alpha smooth actin compared to the BSA. We can see some in the alpha smooth actin, but there is not much difference observed for these E-cadherins. Thereafter they compare these compliant matrices results with these rigid matrices with the similar conditions, that is overexpressing survival factors, that is Bcl-xl. So in these overexpressions, they are able to see increase in the N-cadherins and decrease in the E-cadherin, and alpha smooth actin. And these results are similar to the case where they inject this GFP also, and also where they injected GFP. And for the control, they use this retro. They don't detect the GFP. However, for the compliant case, they did not see any changes. That is, E-cadherins levels remain same, and there is very minimum or almost negligible expressions for N-cadherin and alpha smooth actin. And for the control, they use FDH. Thereafter, they quantify these results individually, and we can see that in the rigid compliant case, they are able to see high expression of N-cadherins when they expressed this overexpressed these survival factors, and for control it they use GFP. However, for the compliant matrices, they do not see N-cadherin expressions, whereas for the E-cadherins, they are able to see high expression levels of these compliant matrices when they overexpress Bcl-xl, or when they inhibit pan-caspase using pan-caspase inhibitor ZVAD. And those signals seems to be higher than the rigid matrices that are treated with a similar thing. However, those differences are not statistically significant, as they are not shown in this figure. But when they check for alpha smooth actin, they are able to see a increase in the signals for in the rigid matrices for GFP, and also for overexpression of Bcl-xl, but not for the case where they overexpress Bcl-xl and use these pan-caspase inhibitors in the compliant matrices in the TGF-β treated conditions. So this in altogether indicate that this EMT and apoptosis are not dependent on each other, as by inhibiting apoptosis they do not see changes in the compliant matrices in terms of increase in the EMT, and the EMT changes results are similar to the case where it did not overexpressed, where they did not treat the cells with TGF-β. There is slight increase, but here the statistically significant results are not shown, or they might not be statistically significant. I did not check that much in detail about the caption. So I hope you understood the main idea, that EMT and apoptosis in TGF-β treated compliant matrices are independent response, as EMT changes does not vary too much when they suppress the apoptosis in these TGF-β treated compliant matrices cells. So that's all for this video. If you need to understand more about the experiments in detail, you can go through the reference that is provided in the description. That's all for this video. Thank you for your time.